Coming to transcription. So transcription can be defined as synthesis of RNA from DNA is called as transcription. So it may be mRNA, rRNA, tRNA, whatever RNA it may be. Whenever RNA is synthesized from DNA, that process we call it as transcription. Right? And coming to phases of transcription, as you see in case of replication, we have initiation, elongation, termination. Same in case of transcription also we have initiation, elongation and termination. These are the phases of transcription. Right? So coming to initiation of transcription, so what happens in case of initiation? So we have to, the initiation of transcription requires a particular recognition of a particular sequence on the gene which has to be transcribed. So we call it as transcription unit. So that transcription unit is nothing but length of the DNA that has to be transcribed. So what do you mean by that transcription unit? It has a promoter region, it has a termination region, and the region which is transcribed. So all this together we call it as transcription unit. And the first step or in initiation, and there is one enzyme called as RNA polymerase. So that RNA polymerase function is it has to recognize a particular sequence, it has to recognize promoter region. So once it recognizes the promoter region, then only it can initiate the elongation. So then only the transcription occurs. So in order to occur transcription, we require the recognition of the promoter region by RNA polymerase. So that RNA polymerase recognizes the promoter region, and for that recognition it requires some sequences. So those sequences in the promoter region we call them as consensus sequences, right? So from the starting point of transcription upstreams, right, that sequence we print as minus sequence. In case of from the starting point of transcription to the upstream, in case of prokaryotes, we have two sequences. Those are minus 35 sequence, and another one is called as Brigno box, that is Tata box. In case of prokaryotes, these two sequences are required for the recognition of the promoter region by the RNA polymerase. Right? So that RNA polymerase binds with that minus 35 sequence, and after that it slides from that minus 35 sequence and it covers the minus 10 region. So where the sequence is TA, AT. So that sequence, up to that sequence it covers, that is covered by the RNA polymerase. So once it recognizes the sequences in the promoter region and covers those two regions, then only the transcription process will be initiated. Right? So the initiation basically involves recognition of the promoter region in the length of the DNA that has to be transcribed. Coming to elongation. So once the promoter region is recognized by the RNA polymerase, so that RNA polymerase actually is a multi-subunit enzyme. It has 2 alpha, 1 beta prime subunits. In addition, that also has 1 sigma subunit. So all these, that's why we call it as a multi-subunit enzyme, because it has 2 alpha, beta prime subunit with sigma subunit. And without sigma subunit, RNA polymerase is called as core enzyme, and with subunit, that RNA polymerase is called as holo enzyme. So that holo enzyme is required for the recognition of the promoter region. It is required for the initiation. So once with that promoter region is recognized by the RNA polymerase holo enzyme, so initially it starts synthesizing some small fragments of RNA, and those fragments will be discarded. Right? And so actually the elongation process will be started. We can say that elongation is started only when a sequence of base pairs like about 10 nucleotides are added, then only you can say that the elongation is started. So that is the significance of sigma factor in case of RNA polymerase, that is required for the recognition of the promoter region in the DNA which has to be transcribed, right? So once that recognition of promoter region is occurred, then what happens then? That RNA polymerase, the sigma factor will be released from the RNA polymerase, and that remaining core enzyme without sigma factor is involved in the elongation process. So that melts the DNA, separate the strands of the DNA. So once the strands of the DNA are separated, it polymerizes the addition of ribonucleotides. Right? So those ribonucleotides are added by this RNA polymerase, and sequentially there is addition of ribonucleotides occurs by the action of this enzyme, and that is complementary to that of DNA strand. So that strand which is copied, you call it as template strand. So in case of transcription, both strands may act as templates, but transcription occurs only on the strand which is recognized by the RNA polymerase, right? So once RNA polymerase recognizes the sequences on the DNA strand, that strand undergoes transcription, and whatever the information in the template strand is there, that is transcribed into the nascent RNA strand. So this is how nascent RNA is synthesized by the action of a multi-subunit enzyme RNA polymerase. So there is simply addition of ribonucleotide triphosphates occurs. So in this addition, pyrophosphate is removed, and that nucleotide monophosphate is added in this addition of in the synthesis of nascent RNA. So this is how elongation process occurs by the action of RNA polymerase. Now coming to termination. So what happens in case of termination? Once the gene which has to be transcribed is transcribed, once the transcription is completed, so at the end of, so as I told you, the transcription unit has promoter region and termination region, and in between that region is transcribed. So whatever the sequence in the template strand is there, that is copied into the RNA, and when it reaches the termination region, the RNA polymerase will be detached from that DNA strand, so that it releases the RNA from the DNA, and again the rebinding of the DNA occurs, and that's how the new RNA is synthesized from the DNA by transcription. So here the termination occurs in two ways. One is row independent termination, and another one is row dependent termination. In case of row independent termination, there is a particular sequence which results in the hairpin formation, hairpin structure formation, that is called as palindrome sequence. So that's how the row independent termination occurs, due to formation of a palindrome sequence. So, and coming to row dependent termination, this row factor binds to the RNA polymerase. So it starts its journey from the 5' end, and once it reaches the RNA polymerase, the function of row factor is, so it detaches the RNA polymerase from the DNA strand, so that RNA polymerase, the termination of the transcription occurs. So briefly you can say that the termination of transcription occurs in two ways. One is row dependent, other one is row independent termination. So this is about the transcription process. So we have discussed about how this how this RNA is synthesized from the DNA, and what are the different phases of transcription, and what happens at the end of transcription. At the end of transcription, you see that we have discussed there are three phases: initiation, elongation, and termination. After that, there is one more process, that is post-transcriptional modifications. Whatever the RNA which is synthesized at the end of transcription, we call it as primary transcript, and that transcript undergoes some modifications, we call it as post-transcriptional modifications. In case of mRNA, the post-transcriptional modification is 5' capping and 3' poly-A tail addition. These are the important post-transcriptional modifications of mRNA, and this is then only the mRNA becomes functionally active. So these four processes are required for the synthesis of a functionally active RNA: initiation, elongation, termination and post-transcriptional modifications.